Manger vos légumes, c'est pas seulement bon pour vous, ça pourrait un jour vous protéger contre le coronavirus. C'est le proverbe de la biologiste végétale Alison McLean qui travaille à l'Université d'Ottawa sur la création d'un vaccin comestible contre le nouveau coronavirus. En outre, sa recherche consiste à injecter des plants de tomates, de laitue et de tabac avec une minuscule particule d'ADN viral nageant dans une solution bactérienne. Nous avons eu l'occasion de rencontrer la biologiste pour en savoir davantage sur sa recherche.
My name is Alison McLean, and I'm an assistant professor here at the University of Ottawa. And I am a plant biologist. And what I normally study is the ways in which plants and microorganisms interact with one another. And so this can both be in the context of disease, so studying the microbes that make plants get sick or get disease. And I study these as a way of protecting plants for farmers so that they can grow better. And I'm also interested in studying the ways beneficial microorganisms can help plants grow more productively in the absence of fertilizers and pesticides. So that's what I normally do. More recently, and the reason why you're interviewing me today is that when the pandemic hit, we were forced to shut down research across the university as part of a way of protecting everybody. And I very quickly asked myself the question, well, what skills do I have as a plant biologist that could contribute to the fight against SARS-CoV-2? And so this is where I came up with the idea of trying to create an edible and more recently an intranasal vaccine against SARS-CoV-2 to protect people against COVID-19.
So what inspired you to do the vaccine?
This was not my idea originally. There is a vast body of scientific literature in which both edible and more broadly plant-based vaccines have been created. And so very much I'm building on the work that has been done by other researchers in this field. Now, what inspired me to do this? Like I said, very simply, I had a certain skill set. I knew that I would be able to have the knowledge and skills necessary to bring this about, and it just seemed a very straightforward thing to try.
So can you tell me what are the potential benefits of having a comestible vaccine instead of one injected by needle?
Yeah, so there's a number of different, the question was, what are the benefits of having an oral vaccine and also a plant-based vaccine versus more traditionally produced injectable conventional vaccines? And so there's a number of different benefits. So firstly, plant-based vaccines can be cheaper and more affordable, both in terms of developing and in doing the research. They can be very quickly to scale up. Once you have something working, you can very quickly scale up and produce large amounts of the protein within a matter of weeks. Also, we aren't reliant as much on what's called the cold chain storage. So this requirement of having freezers or fridges to store your vaccines, that's less of a concern, certainly with the edible or oral-based vaccines. And so we find that oral-based vaccines are really important for developing countries because they are more affordable and because you do not have to have the same infrastructure that you need to produce and also to distribute conventional vaccines. Injectable vaccines, you need to have a skilled, trained workforce to inject these in people in a way that's safe. You need to have sufficient sterile needles available to you. You need to have freezers and fridges to store these vaccines in. And these are all requirements that are not necessary for oral-based vaccines, where you can simply, ideally, provide people with seeds, they grow the plant, and they eat it. As simple as that. Significant advantage that plant-based vaccines offer over more traditionally produced vaccines is that they tend to be safer. So when you're producing conventional vaccines, which are usually generated using eggs, there's always the fear of contamination with the pathogen that can, or something that can cause disease in humans. Because things that can infect eggs can also be things that can infect humans. Now, we don't have that concern with plants because a pathogen that can infect a plant is not going to be able to infect you or I or make us sick. And so this is one less concern we have, and also one of the reasons why it is cheaper to develop these vaccines. So in developing our vaccine, we are using a fairly traditional approach to identifying what part of the virus that we want to use. Typically, vaccines are based on producing exterior bits of protein that are located on the outside of the virus, because that's the part of the virus that your immune system and your immune cells are going to encounter. So in the case of SARS-CoV-2, there's something called a spike protein. This is a protein that is located very much on the outside of the virus, and it's something that your immune system and your antibodies can recognize and help protect yourself against the virus. In particular, we're interested in something called the receptor binding domain. This is a tiny little part of the spike protein, and it's involved with helping the virus bind to your cells and get into your cells. And so the thinking of many vaccine producers is that if we can produce a vaccine that's based on this part of the spike protein, then your body can produce antibodies that will bind to and prevent that protein from working properly. So prevent the virus from getting into your cells and helping your body to clear the virus. And so what we do is we've gotten a company to make us small bits of viral DNA that correspond to that spike protein that we're interested in making. And so then what we do is we produce those bits of DNA into bacteria. These are called agrobacteria. And so these are a strain of bacteria that in nature will infect plants and inject their own DNA into the plant cells, thereby causing disease. Now what scientists tend to do, and what we've known for a few decades, is that we can harness this ability. We can take these bacteria, we can put our own DNA of interest into these bacteria, and then the bacteria will inject that DNA into the plant cells. So in the case of SARS-CoV-2 vaccine, what we've done is we've taken a bit of DNA that corresponds to parts of the spike protein. We've put it into these bacteria. We then grow up large amounts of these bacteria in flasks. We're talking liters of bacteria. And then we basically inject them into the plant. In our case, that would be the lettuce. And the bacteria take the DNA, the viral antigen, and they inject it into the lettuce cells, and then the lettuce cells start to make that viral protein. And the thinking behind this is that when you then eat that lettuce, it will have bits of the viral protein in it. And that's something that your immune system will recognize, will take up and produce an immune response that will help protect you against SARS-CoV-2 and COVID-19.
You mentioned you're in like can you tell us which trial phase you're in?
So we have just finished determining the best combination of DNA, of bacteria, and of plants that give us really nice vaccine, I guess for lack of a better word. And we have tried more than a dozen different plants to bring this about. We've tried collard greens, parsley, spinach, 3 different varieties of lettuce, all kinds of, basically if it has a leaf and you eat it, we have tried to turn it into a vaccine. So we have completed the stage of trialing all these different combinations. We have a really nicely working vaccine, if you will, and we are in the process of giving that to our collaborators, which are at the OHRI, Ottawa Hospital Research Institute, and they will be putting them into mice. So we are entering into the animal study phase of this process.
Awesome. And are you able to tell the success rate of the vaccine? Do you know if like the freshness of the fruit, the vegetable impacts its viability?
We don't know at that point. I don't think freshness should be a major concern. Quite often, edible-based vaccines are lyophilized. So that's a fancy word that basically means you freeze them and then you remove all the water from them. So they're dried. It's like freeze drying, basically. The reason we do that is because things that are freeze dried have a very long shelf life. So they're no longer considered fresh, but they can last for months. And so this is a very commonly used format of producing and storing edible vaccines. It tends to work. It tends to be a safe way of making these vaccines. So I would envisage our vaccine will not necessarily be fresh, and that is an advantage. Fresh things spoil. Freeze-dried things don't. One of the things I'd like to emphasize is that this is not necessarily meant to replace the conventional vaccine. What would be great is if it could be partnered with it. So it could be a booster, for example. So if you get an injectable vaccine, you might need one shot and then you eat something a week or two later and that could be your booster. That might be a really nice way of pairing the two, because injectable vaccines produce a certain kind of immune response in your blood. They help protect you from, it's called systemic immunity, and edible vaccines produce a different kind of immunity, which is called a mucosal immunity. And we think the best way of really protecting somebody against COVID-19 is inducing both types of immunity, blood-based and mucosal-based immunity. And to get that, you need to pair up the conventional injectable vaccine with an edible or intranasal vaccine.
Awesome. And going off that and how you would see it ideally and the logistics, how would it be delivered and administered?
Which one? When you say it, you mean
the comestible one.
The comestible one. What I would envisage would be our lettuce. Right. I think probably the best way of doing this would be for the lettuce to be freeze-dried and packaged into a little tablet. So you would literally just ingest a tablet with a known dosage, and you would eat it with your breakfast, for example. Alternatives would be if we had a line of lettuce that was genetically modified in a way that's very stable, then we could potentially distribute seed to different countries. And this seed, this lettuce, could be grown, and potentially people could just eat the lettuce right there on the spot.
Yeah. How do you feel about those who say the vaccine isn't safe? Or what do you say to anti-vaxxers?
Mm-hmm. Keeping in mind, I am not an immunologist, I am not a virologist, I am not a doctor, I'm a plant biologist. So to some degree, I'm not sure I'm any better qualified to make a comment in terms of vaccine safety than any other scientist. Having said that, I do believe vaccines are safe. I do believe they're important. I will be taking a vaccine. My daughters will be vaccinated. To me, that's a no-brainer. What I would say to people who are concerned about vaccines in general, and also about, there was some concern expressed about my research in terms of it producing a genetically modified organism that is used as a vaccine. And I would say there's extensive data showing both vaccines and the consumption of genetically modified organisms is safe. This is a safe thing. People might argue that this is not natural. What I would say in response to that is that dying from disease is natural. Vaccines in general may not be natural, but they protect you. Electricity, using cars, a lot of things we do are not natural. We do them because they make our life better or they keep us safe. That's what I would say to that.
Pour conclure, le but de cette rencontre avec Alison McLean était de partager avec vous l'ampleur du décor lié à la recherche axée sur le COVID-19. On comprend maintenant l'importance de considérer l'accessibilité d'un vaccin. Pour ce faire, la recherche d'un nouveau moyen d'administration d'un antidote permettrait de mettre fin à une certaine disparité quant à la distribution des vaccins.